Hi guys, I'm Sam from the Unity of Life, and today we're going to be looking at cell division and the principle of animal breeding and genetics, because we have looked at what goes on the outside and all the preparation and all the complicated things that happen when you're breeding your dog. So now we're going to be talking about meiosis, and which is two parts. There's a part 1 and there's a part 2 of meiosis. It doesn't look like we're talking about mitosis, which is weird. But we're going to look at Mendel as well, and we're going to look at some of their laws, and I'm going to try and not make it as complicated as possible. Because there's a lot of things on here that it gives you a little Punnett squares. It shows you how to do the genetics, but there's not an awful lot that I can tell you at the moment without you seeing them. But what I might do is make this into a Google Doc and put the link down below in the description box, so then you can see everything that we are talking about, and you can have a look into it a little bit more detail if you wish. It is the way that it is because it was meant to be a leaflet, so I know it looks a bit confusing because it's meant to get folded up and be like a little booklet. Anyway, so let's get started. So let's have a look at meiosis, part 1. So what happens here? So this is one cell cycle. Like I said, there's meiosis and there's mitosis, but I suppose mitosis is not so much important for when you're sort of breeding your dogs. It is important, but mitosis, I think, is to do with the duplication. No, it isn't. It's just the duplication of cells, where meiosis is the division of sex cells. And I should have remembered that because my biology teacher told Ted and said the way to remember this was GCSE level, is toes aren't sexy. So I suppose you'd think differently if you've got a foot fetish, but anyway. So this is the division of sex cells, which is known as gametes, which is eggs and sperm. And they go through a couple of stages to be able to get the right genetics, and it's a process that everything goes through. So meiosis 1, so this is we've got so the interphase is where a pair of centrosomes, which is found outside the animal cell, each contains a pair of cycloids. Don't worry too much about it, and this is when they're produced. They produce and they duplicate themselves. Like I said, I will put this as a Google Doc. I will put the link in the description below, and we're going to check it out in a little bit more detail, because it is a little bit overcomplicated. So let's have a look at prophase 1. So this is where the chromosomes have these chromonoids attached to the centromen. Bear with my butchering of words, I do that a lot. Meiosis begins by aligning in the homogenesis pairs. I'm sorry, there's a lot of words here that I cannot pronounce because I just can't pronounce them. They're really hard and difficult. And I'm dyslexic, so I can barely speak English, normal English, never mind fancy, toity, technical English, or any other language. So this is where they start to mix up the chromosomes together. So in metaphase 1, because they go through these stages again. So that's when the homoglycis lines up at the metaphase plate, For separation, the orientation of the pairs is random, allowing all the gametes to have different sets of homogenesis. I know I mumbled that, didn't I? Sorry. Just can't pronounce crap. Telophase 1, in the last stage of the chromosomes, are at the opposite ends. Oh, sorry, we missed the stages. The arrows. You go to allophase 1, sorry. And this is the sister chronads of each chromosomes on the homozygous pairs stay attached to each other as the homozygous pairs are pulled apart and now on separate cell sides. Then we go to telophase 1, and this is in the last stage. The chromosomes are at the opposite ends of the cell. In some organisms, this is the end of the gamete division. However, some skip this stage and go into dividing again. So meiosis 2 only happens in certain cells, and it all depends on the cell type and species. So we're going to quickly have a look at that, and then we're going to move on to crossing over, and then we're going to talk a little bit about Mendel and his first and second law. So meiosis part 2. So meiosis part 2 is simpler and shorter than what meiosis 1 is. That doesn't mean I'll be able to pronounce any of the words any better. The cells that were formed during meiosis 1 enter the second stage without copying their DNA. So we still have this prophase, metaphase, anaphase, and telophase. So prophase 2. In this new stage, the nucleus envelope breaks if needed after the chromosomes condense. There are little structures called centrosoids, I think I'm saying that right, that are found near the nucleus. These centrosoids move apart, forming little spindles that capture chromosomes. Metaphase 2, like in metaphase 1, the chromosomes line up individually along the metaphase plate. They do not separate. Allophase 2, this stage is where the sister chromates are split up from one another and are pulled to the opposite sides of the poles. So you could think of it as parents taking one of the children to the opposite sides of the world. They've got two. That makes it a little bit easier to understand. And telophase 2 is each set of chromosomes. They decondense while nuclear membrane forms around them. So meiosis ends with the chromosomes being split by cytosis. Oh, I don't know, forming a new cell. Like I said, I can't pronounce these words. I can only apologise. I am doing my best. These are probably a bad idea to do these videos because I just can't pronounce stuff. But I'm just trying to educate people, and I feel like I'm just this mumbling mess at the moment. Anyway, let's have a look at crossing over. So crossing over is where the parent cells exchange individual chromosomes with each other. This in turn now gives the cells that will eventually finish as the daughter cells a variation of genetic material which would determine a certain feature of an animal, like its eye colour or its fur colour, or our hair colour, because it happens with us as well. So crossing over during prophase 1, each parent cell has a pair of homologous chromosomes. God, I'm butchering words again. Crossing over is important for maintaining genetic diversity because the new cells will have genetic materials that is completely different from both parent cells. This gives the offspring a variety of genetic material and is less likely to succumb to the genetic problems that occur due to the rustle of incest. That's why we don't do incest, people. An example of this is in 2013, it was found that Snowflake I love this gorilla. The world's only known albino gorilla in Barcelona Zoo in Spain had albinoism because it was likely that his mother mated with her uncle. And it was found that his parents actually shared 12% of their DNA. And I actually remember going to Barcelona Zoo when I was really, really young or really little. Snowflake is no longer with us. He died of skin cancer, and I do remember going to see Snowflake, and I was absolutely fascinated with him because obviously he was pure white, while gorillas are generally black. And that's like one of my very first memories, was seeing Snowflake in the zoo. So yeah, Snowflake, I should say, he ended up in the zoo because he was taken from the wild by poachers. Probably because he was an albino and he stuck out, and they were going to try and sell him in the pet trade. But he did go on to have many children himself, and none of them seemed to have albinoism. So that was some good news. So let's have a look at Mendel. So Mendel's really important when it comes to genetics. And there's a lot of videos on YouTube that will probably explain this in much more detail and much better than what I'm going to do. So Mendel was an Austrian priest who conducted a number of experiments with pecan plants. So Mendel's first law, what he did was he basically separated the purple pea plants from the white pea plants. So that was the colour of the petals in the actual flower, not the seeds. The seeds were, I think they were yellow and green. So he bred purple with purple and white with white. So Mendel then took the white pea plants and bred them with the purple ones. He realised that the offspring of the purple and white pea flowers were mainly purple, and that the white ones appeared a quarter of the time, meaning purple was the dominant gene. Mendel came to 3 conclusions. His first conclusion was that the offspring inherited the allele from each parent. This would tell the offspring whether it would be a purple flower or a white flower. So you have these dominant alleles, which means that the offspring are more likely to have the dominant coloration. So darker colours, they tend to be the dominant colorations over bright colorations. So if you've got one parent with brown eyes and you've got one with blue eyes, the likelihood that your children will have brown eyes. You will have the odd one that does, however, have blue eyes. But like I said, I'm going to put this into the Google Doc, and you can see the little Punnett squares that explain that in a little bit more detail for yourself. And I'll put the link in the description below. So let's continue with Mendel. So there would either be a gene from both parents for the colour purple or for white. So the second conclusion was that sometimes there was a gene for both, and the flower with the genes for both would always be purple. As for the gene for purple is dominant, and the gene for the white is recessive. So Mendel's third conclusion was he discovered that the genes from the offspring from each parent was random, even though the purple was being expressed more. So it is interesting to note that everything we know about genetics today is from Mendel's pea plants. However, when he died, he never really published any of his findings because it kind of went against the church, right? Because in Christianity, God created everybody, and everybody is a descendant from Adam and Eve. Where this is proven not to be wrong, and that's a huge no for the church, and all his findings were almost destroyed, but luckily somebody did find them or was able to save them, so we know about all this today. So Mendel's second law. So Mendel took the seed of his pea plants, so the peas, and saw that he had yellow smooth peas and green wrinkly peas. So I was right with the colorations before. From this discovery, he was able to calculate what colour and texture of peas he would gain after breeding them. This from the plant's colour. He also discovered that the genes for the peas and the genes for the plant were not connected but were individual. So, like I said, most of this sort of thing is very, very basic. It's sort of covered most of the time at GCSE levels in the UK. It's something that you all learn at biology. There is obviously a lot more to this, but like I said, I'm going to turn it into a Google document, and I'm going to put the link into the description below, and then you can see all the diagrams yourself rather than inserting them into this video, because that would be overcomplicated. So if anybody's got any questions, drop them in the comments section and I'll try my best to answer them. And I'm only going to answer the questions if they're related to this topic. If they're sensible. If they're not sensible, then I'm not going to answer them. And if you like my content, click on my face to subscribe. You want to. And don't forget to click that notification bell, which then you'll be notified every Wednesday and Saturday when a video appears. And for all my other animal related videos are down here, so you can click on them and check them out. And that is all. Don't forget to share this with your friends if they're interested in this. And other than that, I will see you in the next video. Bye.